Now in this we are going to see how the cell division, first meiosis occurs, then zygote forms, and then how mitosis is taking place of the zygote. Now for example, you just see this is a somatic cell with say human somatic cell with say 23 pairs of homologous chromosomes. So totally there are 46. Okay, this is a 2n. So one particular chromosome, say 6, 7 or 2, some chromosome we have highlighted the homologous chromosome of this 46 of these 23 pairs. One particular pair of homologous chromosomes we have highlighted. So you say one is coming from father and one is coming from mother. Among these homologous chromosomes, it will be one from father, one from mother. So that's why gene locus should be on the homologous chromosome, not on the sister chromatids. This gene locus is from the on the father's homologous father's part of the chromosome, and this is from the mother's part of the chromosome. Okay. Now we know in mitosis, we know in meiosis. Now let us go to page 169. Let us see the pictures in NCERT of the meiosis. You see, this is how it is. These are the, you see, this is prophase, no? This is meiosis 1. So these are the homologous chromosomes, and these are from say one of the parent, and this is from the other parent. So colors are different. Homologous chromosomes. So what is happening? Metaphase, and then you have finally you have a telophase where each chromosome is getting separated. Then coming to the meiosis 2, what is happening? Ultimately, you see each sister chromatid, this is one chromosome, each sister chromatid is coming to the haploid cells. So there are 4 haploid cells. Say these are 4 haploid in the meiosis process, 4 haploid daughter cells, maybe either of the sperm or of the egg. Okay. Now what would happen if they fuse? For that we will see. Now see what we have done. I can cover it this way, I think. See, this is from the sperm, this is from the egg, you think. Same thing we have drawn. Okay. Here the blue is blue and red. Here it is black and plain. Okay. So say this is from the father and this is from the mother. So you have 4 haploids and the 4 haploid. So one of the egg cell will fuse with the sperm, right? And this is going to form the, now note how we have done from father F1 and F2. That is the blue and the red, and the blue and the red, M1, M2. Okay, whatever. See, similar. Blue and the red. Okay, so same thing, blue and the red, like that we have made it. So these two, when they fuse, what is going to happen? See, this and this will come, this and this will come into the cell. This is 2n. Okay, this is 2n. So this is fusion has happened. So you have a diploid. This is haploid number n, means this is diploid number 2n is there, chromosomes. We are talking only about a particular chromosome, say chromosome 6 and 7 or whatever. We are taking about one particular chromosomes pair. Okay, these are the homologous chromosomes. Okay, so now coming to what happens after fusion, zygote is formed. Now zygote has to divide by mitosis. So what will happen is, if you see in mitosis what is happening, page 163, the interface 95% is of the cell cycle is interface. That's why it is divided into G1, S and G2 phase, and after that 1 hour of mitotic phase. M phase is just 1 hour in the 24 hours life cycle. 1 hour only is M phase, baki full 23 hours is G1, S and G2. 95% of the duration of cell cycle. Okay, now the S phase, no, the synthesis phase, what is happening? The DNA amount will double. DNA number will not increase, it's the amount that will double. So that is what is happening in the, see, this is zygote that has formed. Okay, sibling is having like this. Now when it has to undergo mitosis, what will happen? DNA will replicate. See, this was like this, no? Now it has replicated, become double. This was like this, no? Single, replicated, become double. See, each one become double. This one become double. So this is in the, after the S phase, it will be like this during mitosis. Then what happens? Mitosis takes place. Now you can see in mitosis in the page 165 of the NCRT. See what is happening in mitosis. This is mitosis. Okay, mitosis. So we see in the metaphase like this. I mean the red and the blue are each half of divides. Ultimately, what are you having in the anaphase? You are having the sister chromatids half each. You can see, no, like this it was. So it's each sister chromatid is traveling this side one, this side one, that side one, this side one, this side one, this side one. Ultimately, it is like this. Okay. So that is what we have represented here. So now after anaphase, what will happen? This will go there, this half will come here, this half will go there, this half will come here. Similarly, F2's half will go, F2's half will come here. M2 half will come. Same thing, see. M1 half, M1 half, F2 half, F2 half. Similarly, M2 half, M2 half, F1 half, F1. Going to each side. So that now again one division will appear and this becomes a cell, this becomes a cell. So you saw, just made it straight. Okay, so you have it M1, F1, M2, F2. Okay, M1, F1, M2, F2. Okay, so it become 2n. So same thing, just this was bent, I made it, this being made straight, like to show you that it is this is the 2n number you get after mitosis. Okay, now see, the concept of homologous chromosomes is talked about only in meiosis and not in mitosis. If you see in the page 168 in the NCRT, what they are telling is bivalent. That picture, that word bivalent, no, what does that mean? That the chromosomes start appearing as homologous chromosomes. That pairing, no, the complex formed by a pair because of the synaptonemal complex that starts to appear in the zygotine stage, and it will be very clear by the Pacatine stage. So they are talking about this homologous chromosome concept only in meiosis. This is about meiosis stages, the prophase 1 of meiosis, divided into 4 stages: leptotene, zygotine, pacatine, diplotene. 5 stages, sorry. Leptotene, zygotine, pacatine, diplotene, and diakinesis. So in that zygotine stage it is you see synaptonemal complex and you get bivalent, or it's a tetrad because of the homologous chromosome, and it becomes clear by Pacatine stage. See, although short-lived, the next stage Pacatine, it becomes clear. Now clearly appears as tetrads in the Pacatine stage. So NCRT is not talking about homologous concept in mitosis, only in meiosis it is talking about.